Hello and welcome to the latest Technology Networks webinar, Advancing Monoclonal Antibody Stability Studies with Mass Photometry. I'm your moderator, Rory Mackenzie, a Senior Science Writer for Technology Networks, and I'm excited to be here to host today's session. We have a fantastic presenter, Dr. Amy Chow, who will be sharing valuable insights with us today. Amy is a field applications specialist at Refine, a company specializing in the development and manufacture of instrumentation for the characterization of biomolecules using mass photometry. She has a PhD in chemistry from Georgetown University, where she explored protein-protein interactions through single-molecule force experiments, ensemble biophysical characterizations, and computational approaches. I'll now hand over to our speaker, Amy.
Thank you, Rory, for the kind introduction. Hello, everyone. Thanks for attending today's seminar. And I will present two case studies on how mass photometry can be used to assess antibody stability. Antibody aggregation is a major issue in product development, and it is an important attribute to consider throughout the different stages of the development cycle, since it can affect product efficacy and safety. And to understand the physiochemical stability of the candidates, researchers can conduct studies such as force degradation assays. The experiments can include placing samples under different stress conditions and monitor how these samples change. The results provide insights in selecting the right candidates for further development processes. The technology used for these screening assays must be robust and reliable, which at Refine is what we strive to provide to our customer with the mass photometry systems. Mass photometry is an innovative biophysical analysis tool with numerous applications. Essentially, it delivers a rapid, accurate mass measurements of samples at a single molecule level without any labeling. This means you don't need to do a lot of sample preparations before you start characterizing your samples. With mass photometry, you can look at the sample in solution in their native state, which allows you to gain access to subpopulations to determine complex stoichiometry or oligomeric states. This can be of interest if your samples are likely to form different oligomeric states, or if it has any multiple components, the stoichiometry is a useful value to assess. This is also powerful in monitoring macromolecular assemblies if there are any multi-step processes that you want to monitor. Since mass photometry is a single molecule technique, it is identifying individual biomolecules or particles in your solution. It is great at detecting species that are in low abundance or rarely occurring. The last point is particularly important for antibody studies, which is the ability to characterize and monitor sample heterogeneity, providing insights to purity of your samples and addressing stability under different conditions. This is a high-level overview of how mass photometry works. We start with adding a droplet of solution in about nanomolar concentration onto a glass slide. Our system has a laser that illuminates the glass slide from the bottom through the solution. And when it hits the glass liquid interface, the light gets reflected down back to the detector. Some of the light will hit a biomolecule and some of those lights would scatter. And the system will be able to detect both the reflected lights and the scattered light and creates a interference pattern that we can visualize on our software. These interference patterns looks like these radial contrast patterns. And you can see some contrast patterns will be fainter, some will be deeper. And it turns out that the contrast values that we can extract from these patterns are scaled linearly with the mass of the biomolecules, which we can then use and count any of these particles that appear on the screen and plot them as a histogram plot. And on the x-axis, we can convert contrast values to the mass, and on the y-axis as number of counts. And this profile, this mass histogram, allows us to identify the species in the samples and quantify its relative abundance. And so in the first case studies, I will present information from an application note, which is about the characterization of forced antibody degradation with automated mass photometry. This application note is available on our website if you want to read further. The data presented in this case study was part of a broader study that aimed to assess the different effects of the stress conditions. So in here we have light, the presence of hydrogen peroxide, and pH. The study tested 14 different antibodies to establish whether aggregation or fragmentation occurs following the exposures of these conditions. So I will only present 2 representative examples, since 14 is likely going to stretch this presentation a little long. But with 14 antibodies, and if we were to, say, perform a whole experiment, which likely requires running triplicates and any control samples, you can imagine that it will be around 168 total measurements. Well, that sounds like a lot of measurements for a user to manually process and take, and it requires a lot of hands-on time. So with the help of automation, we can achieve 24 measurements in 90-minute walk-away time. This saves users a lot of hands-on time with the instrument. And by removing the human error from pipetting and mixing during those measurements, we can improve the precision of the results. The image on the right here is the inside of our 2MP Auto. There is a tray holder, so the samples are aliquoted in a 96 well plate. The users can design experimental protocols with number of repeats and any dilutions that are necessary. The robot, in this case, performs all of the dilutions in the well plate and takes about 5 microliters of samples to our measurement stage. And the researcher only needs to assemble and change out the glass slide in between protocol runs. So in the first example, we have the mass distribution of each of the stress conditions along with the control. We were not told which of the stress conditions belongs to light, pH, or peroxide, but we can take the histogram profiles and compare the differences of the results against the control sample. The plot here is divided into 3 main areas. The largest peak here that is highlighted in blue is determined to be the intact antibody monomer based on the molecular weight. The region highlighted in orange is considered to be the low molecular weight species, which is likely the fragmented antibody in this case. And the regions that are highlighted in gray on the higher molecular weight ranges are considered to be the aggregations. So in here, for comparisons, we can highlight the same regions across all of the conditions and extract the number of particles detected in them, and to calculate the percentage of abundance. This allows us to identify changes to the sample behavior under different stress conditions, and we can quantify that in the table like this. Where we can see that stress conditions number 2 under the fragmentation antibody column has the highest degree of fragmentation, whereas stress conditions number 2 and number 3 in the aggregations panel has the highest values out of all. And in this one antibody example, 4 different conditions were measured in triplicates, so that totals about 12 different measurements. And with automated mass spectrometry, you can quickly assess the antibody stability with minimal sample preparation and hands-on time. With another antibody example, this experiment was designed and performed the same way as the previous slide. And so here we can distinctly detect not just the monomer peak, but also populations that emerges that belongs to the dimer peak and also the trimer of the antibody. The same analysis can be done just like before, but actually in this case, we can further divide this aggregation region into the dimer levels and the trimer levels. And the values collected is shown here in the table. And by comparing the relative abundance, we can identify that stress conditions 1 and 3 promotes higher dimer and trimer levels, with stress number 1 has the highest trimer aggregation. And based on this information, the users can quickly screen for candidates that are suitable for different purposes. And so overall, in this case study, we show that mass spectrometry is a great analysis tool for rapid repeated analysis for both aggregation and fragmentation. And continuing with the point of aggregations, there are other techniques that are available that can use for like sample characterization workflows. So how does mass spectrometry compare or even complement these solutions? So in this second case study, I will present part of the information from this technical notes on assessing protein samples by size-exclusion chromatography and mass spectrometry. This technical note is also available on our website and goes a lot, it has a lot more detail than what I'll present in this presentation. So this study was completed by RIC Biologics, which is an industry collaborator with us, where they tested 5 different antibodies, the trastuzumab originator and 4 biosimilars. And they wanted to assess the aggregation levels of each of these antibodies. Exclusion chromatography. The sample goes through a column where the species are separated based on the molecular sizes. And so the largest molecules will elute first, followed by smaller species. And the result chromatogram will show on the y-axis UV absorbance of each of the species and is separated by elution time. But with mass spectrometry, the sample is added to a glass slide and the individual molecules are detected and identified based on the mass. So there is no column separation, and what we are getting is just what is in your sample. And the result from the mass histogram will show you the relative abundance of each of the species separated by the masses. And the instrument used for the SEC requires about 10 micrograms of samples, whereas in mass spectrometry, we only need about roughly 30 nanograms. And in this particular study, instead of using the automation like we did in case study 1, this study was performed in our manual 2MP system. And so the user is required to be present with the instrument and pipette the sample onto the glass slide. The users don't need extensive training to be familiar with the instrument. It is very quick and easy to use. From measurement to analysis, users can obtain the results in less than 5 minutes. And so here are the overlaid SEC chromatograms for the 5 antibodies. And the chromatogram shows a very strong mean peak that is identified as the antibody. But if we zoom in to this lower profile here, you can see there are several other peaks that the SCC were able to identify. There is a secondary peak on the left of this monomer peak that was identified as the dimer of the antibody. There are several other peaks that might represent higher order oligomeric states, like aggregation or even fragmentation, or so it's likely coming from contaminants from the purification process. And so the trastuzumab originator peak that is shown in blue, if we look at this bottom panel here, it doesn't have a high abundance of the dimer, whereas clones 3 in red and clones 10 in this olive color have a much higher level of dimer. And the same 5 samples were measured with mass photometry, and the mass histograms were generated, showing here on the right. The blue region was identified as the antibody monomer based on its mass, whereas the orange that is highlighted here in this smaller peaks correspond to the dimer peak. And comparing all 5 samples, clones 3 and 10 have the larger dimer fractions compared to the rest. So it's very similar conclusions that we got from the SCC chromatograms. But in order to have a quantification comparisons, well, we have to first acknowledge that comparing absorbance and number of counts are like comparing apples to oranges. The absorbance data is not only a function of the molecule's concentration, it is also depends on the sample's UV absorbing properties. The specifications of the UV detector itself, like the sensitivity or the at the particular UV wavelength, also influences the data. So we can treat this SCC data by converting the absorbance value that is generated from these chromatograms using the molar extinction coefficients and through the Beer-Lambert's law and the molecular weights of the species to get the relative abundance of this dimer and monomer peaks. And then this calculated abundance value can then be compared to the abundance values with that we get from mass photometry. And so the tables at the bottom here on the left shows the SCC abundance values that have been converted from the absorbance. And then on the right is the abundance values obtained from mass photometry. And we can see that they are in very high agreement, where clones 3 and clones 10 have the highest dimer values. So the results you get from SCC and mass photometry are very similar. And the advantage with mass photometry is that you can get similar results and conclusion with shorter time and less samples. With SCC, each sample uses micrograms of samples and about like 30 minutes per measurement, whereas in mass photometry, it uses much less samples and only 1 minute per measurement and less than 5 minutes of analysis. So if you don't need the separation capabilities that you get from SCC, or you want to be more strategic in selecting the right candidates for these longer separation experiments, mass photometry can help with its low sample consumption and short measurement time. Mass photometry is not only used for characterizing sample aggregation and fragmentation. So in the next few slides, I want to show you some additional applications that mass photometry can be used in the antibody space. So mass photometry can characterize antibody-antigen interactions. In this example, we are looking at interactions between the antibody Herceptin and is antigen receptor HER2. So in this top panel here, we have the control measurements of the antibody alone and also the antigen alone. And by mixing them at different ratios, we can gain insights into the binding occupancy or the complex stoichiometry. And so in this bottom panel here, we can see the binding of either one antigens to the antibody or two antigens to the antibody at a given molar ratio as a mixture. We can explore this further by, say, engineer a mutant version of this antibody and determine how these occupancy changes based on the concentration. So, and this also brings to another aspect of protein interactions, which is to quantify binding affinity. We can determine binding affinity using by doing single-shot KD measurements. So on the left here, we are looking at a glycosylated IgG and its interaction with a Fc receptor. So from the top panel here, we have each of the components measured separately, and the bottom panel is a 1-to-1 molar mixture. And we can quantify the regions that belongs to the receptor alone, the antibody alone, and this complex. So that the regions are grayed out, and we can extract the number of particles in those regions. And that value can be converted into concentrations of each of these components, and then get the dissociation constant. The calculated dissociation constant shown here, it tells us that it has this combination has a very tight interaction. And if we remove the glycan moiety by deglycosylating the antibody, what we get is a very different profile at the mixture. And by performing the same analysis, highlighting the areas for each of these components, converting that value into concentration and get the KD, we can see that it has a 20-fold change in the binding affinity. So this indicates that by removing the glycosylation on the IgG results in a weaker interaction between the antibody and antigen. And so from our two case studies looking at aggregation and fragmentation of the antibodies and the more general applications for mass photometry within the antibody space, this technique is applicable to many biological questions. It is label-free, which allows you to measure the true native behavior of your sample. You can characterize sample purity and aggregation states, looking at oligomeric states, complex composition, stoichiometry, etc. And even using mass photometry as an initial screening tool to streamline your analyses and experiments, identifying the suitable candidates for those much more time-consuming and more expensive experiments. It is quick and easy to use with very minimal sample preparations, and it is cost-effective. Whether it is with the manual system or with the use of automation, mass photometry results can quickly give you insights to your sample, which allows you to strategize these candidates and having time saved on dedicated instruments. And so the two instruments that I've mentioned in this presentation is the 2MP, which is the manual system, and the 2MP Auto with the same internals as the manual system plus a pipetting robot. The manual system can be retrofitted with the automation unit later. So if you don't think automation is necessary at the current stage of the development cycle, you can use the manual system and then upgrade later when the need arises. Refine has been in the market for around 4 years now, and we can proudly say that mass photometry is already making a big impact on furthering understanding of biomolecule behaviors. We have over 170 mass photometers installed worldwide with both academia and industry users. We also have presence in the publication space with more than 150 peer-reviewed papers featuring mass photometry. And we are constantly improving our technology and analysis pipeline so mass photometry can serve our user base better in answering different biological questions. So I hope this presentation gives you some thoughts on how mass photometry can advance your research. So thank you for listening to this talk, and I am happy to answer any questions.